The functional unit of an organ is the smallest structure within an organ that carries out the specific function of that organ. For the kidneys, the functional units are called nephrons. It is the nephrons that produce the urine. Approximately 1 million of these microscopic structures occur within each kidney. Each nephron has two main structures, a glomerulus and a renal tubule. The glomerulus is a highly fenestrated capillary bed. Filtration, the first step in urine formation, moves protein-free plasma from the glomerulus into the second main structure called the renal tubule. Once the protein-free plasma is within the renal tubule, it is called filtrate. Let's take a closer look at the glomeruli. Recall that a glomerulus is a highly fenestrated capillary bed. It is arranged in a ball or a tuft. It is both fed and drained by arterioles. Remember that capillary beds are fed by arterioles and drained by venules. So this is unique. It is fed by an afferent arteriole and drained by an efferent arteriole. In addition to being unique in this sense, the afferent arteriole has a larger diameter than the efferent. This allows more blood to flow in than is being drained at any given moment, resulting in a higher blood pressure within the capillary bed than is normally found. Also, it will allow you to maintain this high blood pressure throughout the entire length of the capillary. This ensures that filtration occurs during the entire length of the capillary. The second main structure of the nephron is the renal tubule. The renal tubule consists of different sections. The section closest to the glomerulus is called the glomerular capsule or Bowman's capsule. It surrounds the glomerulus and consists of two layers. Refer to the figure that I've drawn at the bottom right of this slide. The outer layer is called the parietal layer and is simple squamous epithelium. The inner layer is the visceral layer, and it clings to the surface of the glomerulus. It consists of special cells called podocytes. The photograph at the top right shows you a podocyte with the glomerulus underneath it. The podocytes have processes that interdigitate with one another to form filtration slits. The filtration slits, as well as the fenestrations of the glomerulus, make up the filtration membrane. The space between the parietal and the visceral layers is called the capsular space, and it contains filtrate. This is the protein-free plasma that has been filtered out of the glomerulus. The glomerulus and the glomerular capsule collectively are called the renal corpuscle. Let's continue looking at the renal tubule. The previous slide covered the glomerular capsule. The filtrate now in the glomerular capsule will then flow into the next section of the renal tubule called the proximal convoluted tubule. From there, the filtrate enters into a loop of Henle with a descending limb as we follow the filtrate downward of the loop of Henle, and an ascending limb following the filtrate moving upward. There are several sections within the loop of Henle, a thick section and a thin section. From the loop of Henle, the filtrate then will flow into the distal convoluted tubule. As the filtrate is going through the renal tubule, as mentioned earlier, it is being changed into what will ultimately become the urine. Most of the filtrate will be reabsorbed. Once the filtrate moves into the collecting duct, it will be called urine. What are the components of the nephron? Let's review. The nephron consists of the glomerulus, the tuft of capillaries, and the renal tubule. The different sections of the renal tubule consist of the glomerular capsule, feeding into the proximal convoluted tubule, that then leads to the loop of Henle, the descending limb, and then the ascending limb, and finally into the distal convoluted tubule. Recall that the fluid going through the nephron is called filtrate. The glomerulus and the glomerular capsule collectively are called the renal corpuscle. Notice that the collecting duct is not part of the nephron. There are two types of nephrons. Cortical nephrons have short loops of Henle, and the entire nephron is primarily within the cortex of the kidney. Most of your nephrons will be cortical nephrons, about 85% of them. Their job is to produce urine. The second type of nephron is what we call juxtamedullary nephrons. Juxtamedullary nephrons have long loops of Henle that move into the medulla. About 15% of your nephrons will be juxtamedullary nephrons. Their function is to concentrate the urine. Associated with the nephron is a structure called the juxtaglomerular complex. The juxtaglomerular complex consists of three types of cells. Macula densa cells are located within the ascending limb of the loop of Henle, at the junction with the distal convoluted tubule. These cells are chemoreceptors, and they monitor the concentration of the sodium chloride in the filtrate. The juxtaglomerular cells, or granular cells, are enlarged smooth muscle cells located in the walls of the afferent and the efferent arterioles. There's a higher concentration of these in the afferent arterioles. They measure the blood pressure. In addition, when blood pressure drops, these cells release renin. A third type of cell are the extraglomerular mesangial cells. These cells are located between the macula densa and the juxtaglomerular cells. They contain gap junctions, and it is believed that those gap junctions allow chemical messengers to travel from the macula densa to the juxtaglomerular cells. Associated with the nephrons are collecting ducts. When the filtrate empties into the collecting ducts, it is called urine. The collecting ducts carry the urine toward the minor calyx.